Hello, everyone. For this session, it is our pleasure to have Dr. Yifan Wang sharing his insights about enhanced TCR cell therapy for the treatment of solid tumors. Dr. Wang is the director for target discovery at TScan Therapeutics. He leads TScan high-throughput platform for characterizing the function of T-cell antigen and TCR repertoire for solid tumor therapeutic applications. Prior to TScan, Dr. Wang worked at gene editing companies such as CRISPR Therapeutics and Intellia Therapeutics. He holds a Ph.D. degree in genetics from Harvard University. Without further ado, let us welcome Dr. Yifan Wang.
Okay, thank you for the introduction. Hi, my name is Yifan Wang. I'm a director from TScan Therapeutics. It's my pleasure to be here to be sharing our recent findings with the audience of this conference. Okay, so the title of my presentation is The Discovery of a Novel C0702 Restricted MAGE A1 Epitope and the Preclinical Development TCR-T Candidate for Solid Tumor. So here's our legal statement. I think that's just required. Okay, so let's move to the main topic. So in TCR-T field, we think that this field actually holds great promise for treating solid tumor, but it actually faces two challenges. The first challenge is that only a handful of common targets have been pursued for solid tumor, and the majority of the targets are restricted to one MHC allele, that is A0201. This would actually limit the access for patients for TCR-T-based therapeutics, and it's also relatively easy for the cancer to develop resistance mechanisms by losing the A0201 MHC allele. And the other thing that we found is challenging is that TCRs against common tumor-associated antigens, which are called TAA for short, lacks an adequate safety assessment, which could lead to unwanted side effects for TCR-based therapy. So we developed a proprietary target scan platform, and we think our new platform can be used to address these challenges and bring new therapeutics to patients. So Target Scan is a genome-wide screening platform that identifies cognate antigens of TCR. So this slide describes how the system works. So when you have a T-cell receptor of interest, we basically express the TCR in a large number of T cells and then co-culture the engineered T cells with reporter target cells that also contain a genome-wide protein fragment library. So in each target cell, it contains protein fragments that are 90 amino acids long, that are the components of the library. And the 90 amino acid protein fragments were processed into short peptides, short epitopes that were presented, that were processed by the proteasome and eventually be presented by the class 1 MHC complex. And then during the co-culture stage, when a certain target cell that it can express an antigen that can be recognized by the TCR of interest, this would trigger the T cell engagement of that target cell. And in our reporter cell, in our target cell system, there's also a reporter construct that is an infrared fluorescent protein, which is activated by granzyme B, activation. So when a T cell engages a target cell, it actually secretes granzyme B into the target cell, and this would activate the IFP signal in the target cell. So we developed a very efficient cell selection approaches that could allow us to sort out the cells that were actively targeted by the T cells, and then the active sorted cells would then be sequenced for their library content. So if you are a target cell that encodes a 90-mer protein fragment that actually contains the antigen of that TCR, this protein fragment would actually be enriched during the screen. So in this way, we can identify cognate antigens of TCRs. Okay. Another key component of our system is a genome-wide protein fragment library. As you can see here, the library covers the entire human proteome and is made of overlapping 90 amino acid protein fragments. So the current size of the library is higher than 1 million constructs, and the library is made of various components. All of the non-mutated protein fragments encoded in the human genome are included in the library. And in addition, we're including additional categories of antigens as well. For example, we're including endogenous retroviruses, non-coding ORFs, misspliced RNA species, shared neoantigens, and oncoviral antigens, as well as common SNPs too. So this would greatly extend our search space for finding new epitopes for TCRs for therapeutic application. And then using this target scan platform, our strategy is that to study checkpoint therapy responders to identify targets and TCRs that can be used to help new patients. The thought is that checkpoint therapy has shown great promises for treating solid tumor, but the issue is that most patients don't have robust response to this therapeutics, largely because they don't have an endogenous T cell pool that's potent enough to unleash a full-scale clinical response against solid tumor. But what we can do is that we can interrogate the patient's T cell repertoire who have responded to the immuno checkpoint therapy very efficiently to identify what TCR and antigens are driving the clinical response, and use the information and use the TCRs that we uncover from the responders to make TCR-T therapeutic products to help those patients who aren't winning their fight by using the ICB checkpoint scheme. So here is one case that we recently completed with our collaborators at Dana-Farber Institute. So we're using clinically active TCRs identified from head and neck cancer patients who are responding to immunotherapy to identify new TCR and targets. So the left-hand side describes the scheme of a trial that was recently published by Dana-Farber. So a bunch of head and neck cancer patients either received anti-PD-1 immunotherapy or anti-PD-1 in combination with anti-CTLA-4 for treating the head and neck solid tumor. And then once the patient receives the treatment, after a few weeks, the tumor was surgically resected. So this is a kind of a neoadjuvant scheme. And then the unique factor about this study is that we actually had access to pre-treatment tail biopsy, and also we have the post-treatment surgically removed tumor sample, and this would allow us to interrogate the TCR frequencies and the single T cells that we can identify from both before and after the treatment. So on the right-hand side, we can see that here's an example of a patient who responded well to the immuno checkpoint therapy. You can see that their TCRs have become expanded during the treatment. For example, the red lines are TCRs that can be found in the pre-treatment tumor biopsy, and after the treatment, you can see that this red line TCRs, their frequencies become increased. And in addition, there are also a group of TCRs that were below the detection limit before the treatment, and then they become more abundant during and post the treatment. So basically, we focused on a bunch of orphan TCRs that seem to become clinically expanded as a result of the ICB treatment in those responders. And here's what we have found. So we found that we can use our multiplex, the target scan screen, to identify new epitopes. And then, from this screen, we identified MAGE A1 as a target of the expanded TCRs from an immunotherapy responder. Here is a case for patient C0732. In this patient, the patient had 60% reduction in primary tumor size following anti-PD-1 and anti-CTLA-4 therapy. And here's the screen that we performed to uncover new targets. So essentially, 10 orphan TCRs were screened together with one control TCR. And we used this pool of 11 TCR and screened it against a target cell line that expressed 6 MHC alleles that were that matches the allele, the class 1 allele profile for this particular patient. And from the screen, we identified 2 new targets, and the 2 targets are MAGE A1 and sonic hedgehog. And MAGE A1 caught our attention because MAGE A1 was highly expressed in several solid tumor types, and is also minimally expressed in most normal tissues. As you can see that MAGE A1 is strongly expressed in melanoma, head and neck, lung cancer, and also cervical cancer. In the normal tissues that we looked at, in most of the tissues, the expression level is very low, except for testis. So MAGE A1 is also known as a common, as a cancer testis antigen. So because we identified this screen kit from a multiplex screen where multiple TCRs and the MHC alleles were involved in the screen. So what we did next is to do deconvolution studies to figure out the scoring 90-mer protein fragment to figure out which MHC allele would present the epitope that were processed from that scoring protein 90-mer fragment. So as you can see here, we run the deconvolution studies where we either express all 6 patients MHC class 1 genes, or we express individual MHC class 1 genes. As you can see that when we co-cultured the T cell pool with reporter cells expressing MHC C0702 plus the MAGE A1 90-mer, it caused T cell activation as is identified by the activation of the IFP coenzyme reporter. So now we know that the new target is restricted to MHC C0702. And on the right-hand side, you can see that the allele frequency of C0702 in the US is about 24% in the total population. The next slide shows that because we have completed the deconvolution of the MHC gene. The next step is to figure out which TCR in the T cell pool that were used in the screen were responsible for activating this hit in the context of the screen. So we expressed each TCR from the pool individually in CD8 cells, and then test them against a reporter cell line expressing the MHC allele C0702 plus the MAGE A1 90-mer protein fragment. And surprisingly, there are actually 2 TCRs that were causing T cell killing in the context of the screen, as is measured by the IFP coenzyme reporter activation. And then, so what this means is that in original patients, there are 2 different TCR that were recognizing the same target gene. And because we already know that the target is a peptide that's restricted to the MHC C0702 gene, so that means that there's at least one novel T cell target from this MHC allele. And we know that the C allele covers about 24% of the total population in the States. And then on the lower right-hand side, you can see the profiles of the 2 TCRs that we identified from the screen. TCR39 was actually observed to be quite abundant in this post-treatment patient. It was corresponding to more than 1% of the total kills in the patient. And then the other TCR41 was also quite potent as well. It was actually expanded for more than 10-fold during the treatment, according to the study. And this slide is a summary of the epitope that we were able to map from the 90-mer protein fragment. And you can see that on the right-hand side is a dose curve using the mapped peptide. So the epitope for this MAGE A1 target is VRFFFPSL, and you can see that the affinity of the TCR of the two TCRs to this epitope is actually quite high. So we think that the affinity is in the range of about 200 picomolar. And then on the right-hand side, you can see that is the outline here is the predicted binding affinity of this epitope to the MHC C0702 complex. And you can see that the predicted affinity is actually quite low. So this means that using our experimental approach, we can identify novel biologically active TCR and epitopes that would otherwise be missed by bioinformatic approach. And then because we have found two TCRs for this target, we later choose to nominate TCR number 41 to be TSC204C7 as our new solid tumor program's candidate therapeutic TCR. And then the next study we did is to look at the in vitro efficacy of our new therapeutic TCR TSC204C07. As you can see that we tested our TCR's cytotoxic profile against 3 cancer cell lines with varying degrees of MAGE A1 gene expression. As you can see that it consistently kills cancer cell lines very efficiently. And then this story is actually part of a very recent publication on Cell. The title is provided down below. So if you are interested in the story, please look it up. And then the next step we did is to show that whether our TSC204C7 TCR has activity in a in vivo model setting. So here we developed a mouse model using a cancer cell line that expresses both MHC C0702 and the MAGE A1. Then we dose the patient. Then we dose the mouse with control T cells or with our TSC204C07 product. And as you can see that by injecting the 204C07 T cells into the mice, it greatly reduced the tumor progression in the mouse. As you can see that the red line is much lower compared to the control black line. And on the right-hand side, you can see the behavior, the measurement of each individual mice in the study. So here, basically we're providing an example where we use target scan to identify new targets and new TCRs. So far, it's been very encouraging. And the other thing I mentioned early on is that we're using the target scan platform as a safety assessment tool. So the reason that prompted us to think, to use our platform also as a safety scan tool is that we know that previously there's been some reports that there are certain fatal side effects in the previously tested TCR trials. I think one sad example is that a few years ago there's been a MAGE A3 melanoma TCR trial where people were using an affinity-enhanced MAGE A3 TCR for treating solid tumor. And then, unfortunately, during that trial, several patients developed cytotoxicity, cardiomyocyte inflammations in their heart, and several patients were actually killed by that therapeutic TCR. And we knew that that TCR was an affinity-enhanced MAGE A3 TCR because using the target scan platform that we have, we can basically uncover many unknown off-targets for any therapeutic TCR. So we wanted to ask the question: can we use our platform to identify relevant off-target genes for known side effects of TCRs that have been tested previously? So here we actually use our platform to run the safety profile scan for the affinity-enhanced MAGE A3 TCR that was used by another company. As you can see here, we identified a very large number of potential off-target genes that can be recognized by this TCR. And you can see that the titin was actually rediscovered using our platform as well. And then, more surprisingly, there are actually many other genes that were previously not known to be recognized by this TCR as well. And then many of the newly identified off-targets share some level of sequence similarity to the on-target peptide, but the degree of the similarity is actually quite low. So it's actually not surprising that previously, when people run bioinformatics pipeline, some of the off-targets were actually not picked up by the analysis. So I think this shows an example where we can use the target scan platform to identify relevant off-target genes. So next, we wanted to do is to look at our new TCR to see what is the off-target profile is like. So here we run the target scan safety screen to test our TCR 204 C7, and you can see that from the screen we actually identified 7 potential targets of our TCR. So the next stage is to vet these potential off targets to see if they can really be causing concerns in our pipeline. So here is the summary of the work that we did. So first, because the off targets that we identified from the screen, they were scored in the format of anti-tumor protein fragments. So the first thing we wanted to do is that can we validate the anti-tumors that's scored in the screen? So by doing this, we're able to rule out 4 genes that they actually cannot be validated when we re-express the anti-tumor in the target cells. And the next question is that because of the way the screen system works, we were only testing the antigen reactivity by expressing anti-tumor protein fragments. Then the next thing we did is to express full-length WRFs that were from those identified genes to see whether the potential epitopes can also be produced by expressing the full-length WRFs, and it turns out that 3 out of the 4 genes cannot cause T-cell killing when you express the full-length WRFs. So the only thing that remains to be addressed is one gene that's called LAMB4IR. So the LAMB4IR stands for LAMB4 intron retention. So this is a construct in the library that's cloned into the library because it's part of an intron retention version of the it's an intron retention version of the transcript of this gene. So it can also cause T-cell killing when we express the full-length intron retention WRF. But when we tested the antigen engagement when we pulse the peptides of this LAMB4 intron retention peptides and then measured T-cell killing and compared that to the killing mediated by the MAGE A1 peptides, and it turns out the affinity of the TCR to LAMB4 is much weaker compared to the affinity of this TCR to MAGE A1. So it means that the TCR preferentially binds to MAGE A1, and then the affinity of this TCR to LAMB4 is actually really weak. And then on the other hand, the LAMB4 intron retention transcript is actually very, very rare in normal cells. This intron retention happens at an extremely low frequency, so we don't think that the LAMB4 is of any major concern for us. So the next thing we wanted to see is that we want to still see if the TCR 204C7 can cause cytotoxicity when co-cultured with normal cells. So here is a part of the data where we co-cultured TCR 204C7 against 3 normal cancer cell lines, and we found that there's no cytotoxicity when we set up the co-culture experiment. And then the next thing we wanted to address is to see whether TCR-204C7 has any alloreactivity to the other class 1 MHC genes from the general population. So we set up an array screen where we express single MHC alleles in HEC293 T cells, and then we generated a whole library of reporter cells that in total express 110 MHC alleles, and then ask, does our TCR cause killing in any of the MHC expressing cell line? And it turns out that this TCR shows basically no alloreactivity to any of the 110 MHC alleles that we tested. So it seems to be relatively safe with regard to alloreactivity. Okay, and currently we are developing a new transposon vector for TSC204C7, because one of the challenges for the clinical application of a TCR-T product for treating solid tumor is to face the challenge of the hostile tumor microenvironment. So we are introducing two elements into our vector for expressing TSC204C7 in T cells. One is that we're co-expressing CD8 co-receptor in our vector, and this would allow us to utilize the TCR to express it in both the CD8 cell compartment and also the CD4 cell population. And we hope that this could allow us, the cytotoxic CD8 cells to get the cytokine help that are expressed from the CD4 cells when the TSC204C7 engages cancer cells in the CD4 cell context. And then the other thing we're currently evaluating is to express a dominant negative TGF-beta in our vector. So this would allow our cell therapy product to be resistant to the inhibition of that is caused by the soluble TGF-beta ligand that the cells can encounter in the solid tumor microenvironment. So we believe that using the new construct, we can actually make not only the T cells that will target tumor cells, but that they can also persist well in the solid tumor microenvironment. So here's an overview of our strategy. So TScan's mission is to apply very effective TCR-T therapeutic options for multiplex solid tumor therapy. So what we want to do is to basically extend our pipeline and to be able to target multiple targets for treating solid tumor. So far, we have announced that we will have an HPV program, and then we're currently also setting up the work for the MAGE A1 and the PRAME program. And what we hope is that for each solid tumor target, we can target the gene by using TCRs that are not necessarily limited to just one MHC allele, that is AO201, but also we want to be able to have multiple TCRs that can allow many new patients to have the access of TCR-T therapy. So as a result of that, so at this point we are setting up the work to file for the IND for the MAGE A1 C07 program. But currently we also have an AO201 based MAGE A1 TCR that are currently being evaluated in our pipeline. And then we look forward to the next year where we'll have actually a few more INDs. So eventually we wanted to be able to cover multiple MHC alleles and multiple targets, and then this would enable us to perform multiplex TCR therapeutics for treating solid tumor. So that's it. Thank you very much for your attention.